Welcome back to another fun-filled lecture. I'm Shay Spencer, actually not Clint Hackney, but this is the beginning introduction lecture to the cellular level of organization. I'm going to go over some key terms, and these are going to be important terms in order to understand where we're going with this chapter. So you need to understand that the cell is the basic structure and functional unit of all living organisms. I think that has been a test question ever since you've been in the ninth grade. Uh, it shows up all the time. There's two different types of cells that we primarily talk about: eukaryotic, meaning the true cell, and prokaryotic. Eukaryotic differs from prokaryotic in the fact that it has a membrane bound organelles and that it has a nucleus. So those are that's a difference between the eukaryote and the prokaryote. Then we have something called the cell doctrine. What is the cell doctrine? This is something that was discussed in Bio 112, your intro class. This is just saying what we simply said above, that the cell is the basic unit of life, that new cells come from old cells, right? They come from all pre-existing cells. And you guys already know about that because that's what mitosis is. Mitosis is one cell undergoing division to produce two cells, and then two cells producing four cells, and four cells producing eight cells. And then this third tenet says here it's the fundamental unit of structure, physiology, and organization, right? So that's called the cell doctrine. So it's a basic unit of life. All cells come from pre-existing cells, and it's the fundamental unit of structure, physiology, and organization. Then we said down here, talking about cell division, this is that all cells arise from pre-existing cells. What is important for examination purposes is knowing these different organelles, knowing the plasma membrane, the cytoplasm, and the nucleus. That's going to be primarily the three main components that we're going to discuss. I want you to think of the plasma membrane as being a doorway. This allows passage of molecules into and outside of the cell. It separates the cells from the internal environment from its external environment. It has a flexible outer surface that acts as a selective physical barrier, and that's going to be important when we start talking about things like permeability. Permeability meaning the ability to enter in or out of the cell. Then we have the cytoplasm. Once you think of the cytoplasm as the fluid in between the membrane, the cell membrane, and the nucleus. So you've got a plasma membrane, that's your door, that's your doorway. You have the cytoplasm, this is your fluid portion, and then you have the nucleus. And the nucleus, everybody knows, this is where all of your genetic DNA is going to be contained. Speaking about the nucleus, we know this is where DNA is housed. If I were to ask you in which of the following organelles is mitosis occurring, what would you say? Well, the answer should be nucleus as well, right? Remember, during interphase, we have G1 moving to the S phase. What does the S phase stand for? That's DNA synthesis, absolutely. So DNA synthesis is actually occurring within this nucleus, and mitosis is actually occurring here as well. DNA, right, through mitosis, the formation of the double helix, eventually will be wound into something called chromosomes. And DNA plus a protein is going to be called chromatin. So, chromatin. So, this chromatin is packaged into these um, um, structures called chromosomes. And then, chromosomes we know is the unit that encodes genes. So, I want you to think about it in terms of increasing complexity. So, you have the DNA double helix. From there, it associates with a protein. We know that's called chromatin. The chromatin becomes compact, forms a chromosome, and the chromosome comes together to form genes. This is a very good picture to be familiar with. This is going to be all of the organelles inside of the eukaryotic cell. Here, we'll just talk about the three that we just talked about and then hit on a few. This is the cell membrane, as you can see here. This is enclosing all of the cell. This is going to allow entry and exit of material inside and outside of the cell. Inside here, this fluid portion in between the cell membrane and the nucleus being over here in this purple area, this is going to be the cytosol. So this is the fluid portion. And as you can see back on the previous slide, there was terms called organelles. Everything that exists inside of here, those are called organelles. These are just going to be compartmentalized structures that have specific functions. Like we talked in lab that the Golgi apparatus was essentially like FedEx, right? This was the shipping center of the cell. So again, just touching on the cell membrane, this is the passageway, the door that separates the internal environment from the external environment. Then we have the nucleus, which houses the genetic material. This is where mitosis occurs. This is where DNA is packaged into with proteins to form chromatin, and chromatin is packed even tighter to form chromosomes, and eventually those come together to form genes. And then we have the cytosol, which is the fluid portion that contains all of the organelles. This is just another picture here depicting the nucleus a little bit more. This outer envelope, this is the nuclear envelope. This is an outer protective layer that separates um, the nucleus from the outside. It essentially acts like its own cell membrane right here. So it has an outer membrane, this lighter yellow portion, and this darker inner membrane. You have something called the nucleolus. The nucleolus is going to be responsible for producing ribosomal RNA. And then we have that chromatin that we previously just talked about. And chromatin is essentially just DNA plus a protein. So DNA plus a protein. These nuclear pores, this allows entry and exit from inside of the cell. This is a picture just again depicting what I previously talked about. Here you have the DNA double helix. It's associated with proteins. These proteins are called histones, form something called a nucleosome. But for our purposes, we are talking about the DNA double helix here. Associates with proteins. Then it comes together to form chromatin, right? And then chromatin will eventually become chromosomes. And then chromosomes will eventually form the unit of chromosomes themselves will encode genes. Again, just another picture kind of depicting what we previously discussed. So this the plasma membrane, we're going to, I gave you a brief overview. Now we're going to kind of get a little bit more in depth, beginning talking about the plasma membrane. This is an important structure, right? Like I said previously, this is the doorway. This allows entry and exit into and outside of the cell. The plasma membrane is going to move through something called the fluid mosaic model, and that's an important term to be familiar with. Test writers all love to ask. They like to discuss about the plasma membrane, which of the following describes the movement of the plasma membrane, can be best understood by which of the following, those types of questions, and it would be the fluid mosaic model. Um, so the plasma membrane is a flexible and sturdy barrier that surrounds the cytoplasm of the cell, and we previously just talked about that. Here is going to kind of depict this fluid mosaic model. This is going to be the inside of the cell. This is going to be the outside of the cell. And this just goes to show you the fluidity, how it likes to move. Um, it's kind of counterintuitive. You have this cholesterol, which is present inside of the cell, which helps provide some rigidity. But the phospholipids are able to interact and move to provide that fluidity. And the fluidity of the cell membrane is very important. It helps maintain the integrity of the cell membrane. It allows for proper movement, things like that. Also within this picture, you're going to see the phospholipids. Remember that the cell membrane consists of a phospholipid bilayer. You're going to see the polar head here, which contains phosphate, and the hydrophobic tail or the non-polar tail. And this is going to be consisting of fatty acids. This is a lipid bilayer, meaning two layers of phospholipids. Again, you have a hydrophilic head here, which is polar, and then a hydrophobic tail, which is nonpolar. So again, we've talked about what the cell membrane is. It's a doorway. It allows entry of things into and outside of the cell. We've talked a little bit about the structure. I want you to think of the structure as primarily phospholipid bilayer. That's whenever somebody says the cell membrane, you should always think phospholipid bilayer. But now we're going to start talking about these proteins that are also present within the cell membrane themselves. So there's different types of proteins here. You have a peripheral protein, as its name implies, it's on the periphery. And that kind of makes sense if you think about it. It's on the periphery, so we'll call it a peripheral protein. What is a new word to you guys is something called an integral protein. And I want you to remember this one, an integral protein. This essentially is a transmembrane protein that spans both layers of the membrane. And this is going to be important for functions like ions. This is going to allow ions to be able to move into and outside of the cell. Cholesterol, this cholesterol here is going to help maintain stiffness and rigidity. Um, so here, let's get into a little bit about it. Um, so the plasma membrane, like we previously just said, is a flexible and sturdy barrier that surrounds and contains the cytoplasm of the cell. We know that. It's described using the fluid mosaic model. And again, this is just how the plasma membrane likes to move. Here I have the membrane lipid molecules exchange places 10 million times per second. Wow, 10 million times per second. That's completely around the cell in 2 minutes. Within this lipid bilayer, you have different things. Phospholipids, we know that there's a polar head and a fatty acid tail. And then we say that we have cholesterol. And again, cholesterol increases the membrane rigidity, right? It maintains the stiffness or the integrity of that cell membrane. Note, the bilayer arrangement occurs because the lipids are something called amphipathic. Amphipathic. And that is because the polar part is phosphate containing, that is hydrophilic. I want you to think of phil meaning an affinity for, and then hydro meaning water. So it has an affinity for water, right? So when you kind of look at this, I like to think of this as uh, this is not afraid of the water, but the tails, they're trying to get away from the water, right? They're pointed away. So this has an affinity for it. It doesn't mind to be out near the fluid, but the tails, they're afraid, so they're kind of pointing away. Then the nonpolar parts are long fatty acid tails. That's the hydrophobic portion. Again, this is going to be a slide that just kind of shows you the structure on a more deeper level. This is the phosphate head. This is the fatty acid tail. And again, you can see that here depicted. It's the phosphate, the fatty acid tail. That's in this cross section, this is what you're seeing. So we've talked about the phospholipid bilayer. We've talked about the phospholipids. Now let's talk about the proteins. Remember I said that there's a few different important proteins. You have the integral, which are trans, meaning on both sides, membrane. Transmembrane proteins. These span the entire lipid bilayer and protrude into the extracellular fluid in the cytosol. And an example of these is transport channels. Let's go back up to this picture and look and see what we're talking about. This is an integral protein. This is spanning the entire membrane right here. Do you see this? It's going through both sides. And you can kind of imagine that an ion, something like chloride or sodium, something like that. Remember, ions are molecules with a positive or negative charge. They dissociate once they're in water. These would go through this transmembrane channel right here. Peripheral proteins, again, these are going to be on the periphery. These are associated loosely with polar heads and membrane lipids or with other integral proteins. They're found on the extracellular or the cytosolic side. An example of this would be a glycoprotein. This is just a protein with a carb. It's used to attach to things. This is a picture to be familiar with. I like this picture. This is just on a simplistic form. This is your cholesterol. What does cholesterol do again? That's right, it maintains the stiffness and the rigidity for the cell. This is the transmembrane, and we know it's transmembrane because why? That's right, it's spanning the entire portion of the cell. And this is the peripheral protein on the outside, right? A this is going to be a protein attached to a carbohydrate or a lipid attached to a carbohydrate. This is going to be able to attach to things. And again, this is the phospholipid. This is going to be the polar head containing phosphate. This is going to be the hydrophobic nonpolar tail containing the fatty acid chain. These are just some functions of the protein of the membrane proteins that I've listed. Ion channels, I think, would be one to know. There are selective carriers, but for our purposes, let's just primarily focus on the ion channels. So this gets into a discussion about permeability. So permeability just means what can pass. If it's permeable, it means it can pass through. If it's impermeable, it can't pass through, right? Selective permeability means what? Some things can pass, some things can't. So when you think about things that can pass through the cell membrane, I want you to think of two things. Small things can get through. Think of a doorway. If the doorway is made for a 6-foot man, a 7'2" guy can't pass through, right? And that just means only smaller people will be able to get through that. Only people with certain size. And then I want you to think about polarity, right? Non-polar things are going to be able to get through. And the reason why they're going to be able to get through is all due to charge. You have polar hydrophilic heads, which are going to allow nonpolar things to come through. If you remember, opposites attract. That's very true. Whereas if you had polar molecules coming through, those are not going to be able to pass as easily. There's a few of these that I think would be important to remember that are able to pass through. I think oxygen is one. If you think about the molecular formula for oxygen, that's just O2, or two atoms of oxygen bound together. So that's a small molecule that should be able to get through. Think about carbon dioxide, that is CO2. That's just three atoms bonded together. That should be able to get through. Steroids can get through because they're lipophilic, they're made of lipids, therefore they're nonpolar and are able to easily get through. Things that are impermeable to ions and large uncharged polar molecules. So glucose is a large molecule. Glucose is not going to be able to get through on its own. It's going to have to require something. Calcium. Calcium is going to be polar. It's not going to be able to get through. Something that makes a difference, if you remember the discussions from Bio 112, we talked about something called concentration gradients. This just means how much is inside the cell versus how much is outside the cell. Depending on the steep of the concentration gradient, for example, if you have 10 molecules on the inside versus one on the outside, where is your concentration gradient going to, where do the molecules want to go? Well, they want to go to the outside. Where there is more of something, it wants to go to an area where there's less of something. So the definition of concentration gradient is just the difference of concentrations from inside the cell to outside the cell. The cell utilizes this free kinetic energy. If you remember what kinetic energy is, this is just energy in motion to conserve its own ATP. This is just a picture here that I think is very good. This is essentially just going to show you what can get through. And again, small hydrophobic. Hydrophobic just means what? Non-polar, right? Polar molecules are hydrophilic, whereas non-polar are hydrophobic. So oxygen can get through, it's small. Carbon dioxide can get through, it's small. Nitrogen can get through, it's small. Water can get through, it's small and uncharged. So now we're going to begin talking about simple diffusion and different modes of transport. So again, we've talked about the plasma membrane. We've said that it is a phospholipid bilayer, right, with hydrophilic heads containing phosphate. That portion is polar. It's hydrophilic. It means it loves water. It's polar. Whereas you have hydrophobic tails, right? Those are the fatty acid portion. We said that small, uncharged molecules or nonpolar molecules can get through. And we said that large charged molecules can't get through. We've also said that concentration gradients help us to get through. We've talked about the different proteins that help make up the uh, cell membrane. We've talked about the integral proteins, how those span the entire membrane and allow passage of molecules through. And then we've talked about peripheral proteins, which help attach to things. Now let's talk about how do things actually get across. This will take you back to Bio 112 and some of your high school biology classes, if you think about it really. There's something called simple diffusion, and this is passive. What does passive mean? Does it require energy? No. So passive processes do not require energy. There's no ATP involved, right? Does it require a carrier protein? No, it doesn't. It's passive. It doesn't require a carrier protein. So, and I want you to think about it going from a high to low gradient. So again, simple diffusion, this is a passive process where the random mixing of particles occurs because of kinetic energy or energy in motion. Two definitions to be aware of, and you guys already know this. Solute is just the dissolved substance, whereas a solvent is the thing that's dissolving it. So if you think of salt water, water is the solvent, and the solute is going to be the sodium chloride, right? The salt. So we said that solutes will move down their concentration gradient. If I have 10 people on one side of the classroom and I have one on the other, the concentration gradient is going to favor more people going to the other side of the classroom because there's less people, right? There's more space on the other side. Things that can help increase this, one I think you should know is the steepness of the concentration gradient. If I have 50 people on one side of the classroom, there's a chance that more people are going to move over to the other side of the classroom. We just have more people. Makes sense. The temperature, you increase the temperature, you're going to move quicker. There's more collision of particles. The surface area, the diffusion distance. If there's a smaller distance for the molecules to travel, of course they're going to get there quicker. And then the mass of the solute as well. Wrapping up, we'll talk about facilitated diffusion and then we'll end this lecture. Facilitated diffusion, I want you to think of it in these terms. Facilitated diffusion is just like simple diffusion, right? But it requires a transport protein. Again, I'm going to repeat that. Facilitated diffusion is just like simple diffusion, but it requires a transport protein. What do you mean, Shay? Okay, for example, simple diffusion is passive. It means what? It doesn't require energy, right? Simple diffusion is passive. It does not require energy. Well, you said that simple diffusion is just like facilitated diffusion, Shay. Except facilitated requires a protein. Well, that's right. So facilitated, therefore, must be passive. Facilitated diffusion requires no energy. Simple diffusion goes down a concentration gradient. Guess what? Facilitated diffusion goes down a concentration gradient. So why does facilitated diffusion require a transport protein? Well, when solutes are too polar or highly charged, they use an integral protein. Well, what's that integral protein? Remember, that's just that protein that spans the membrane. So remember how I said ions can maybe go through a channel? That's exactly what I was talking about. There's two different types. There's channel mediated and carrier mediated. A channel just allows things to go through, and a carrier carries it over. That's literally all that is. When a solute moves down its concentration gradient through a transmembrane channel, carrier mediated. When a solute binds to a carrier protein on one side of the cell and it's released on the other, it will bind more often on the side with a higher concentration gradient. This is a picture depicting what I've already said. This is simple diffusion. Let's look at the bottom. Passive transport. All of these are passive, right? I said that facilitated is just like simple. It is. Passive transport requires no energy. This is simple diffusion. Do you see a transport protein present? No, doesn't require one. It's going from an area of high concentration to an area of low concentration. Again, this is passive, going from an area of high concentration to an area of low concentration. What's the difference? This has a channel. This is a type of facilitated diffusion using a channel transmembrane protein, right? It's a transmembrane protein. And guess what this is? This is using a carrier protein. What's the difference in the channel and the carrier? One's getting carried over, and one is just passing through the channel. It's essentially all this is, guys. This transmembrane protein, again, just showing you facilitated diffusion, how the transmembrane protein is facilitating entry of some nutrients. The example that text is using here is fructose. It's going from an area of high concentration to an area of low concentration. That is going to wrap up this first lecture preview. Again, if you have any questions, reach out to me. These are concepts that are not too difficult. Sometimes they can feel intimidating at first, but I really think if you just sit down and kind of look at the differences between simple and facilitated, understand the basics about the organelles, what are they responsible for doing, what's unique about them, different things like that. Understanding that the cell membrane moves through the fluid mosaic model, that's all important for this first lecture preview. So, really enjoyed my time with you guys today. If you have any questions, please feel free to reach out.